Hello, everyone. I'm going to be talking to you today about the association between Drp1 mediated mitochondrial fission and the regulation of cancer stem cells. So first, I wanted to talk to you about the mitochondria. It is known as the main source of energy production for the cells that depend on it. It also has two membranes, each associated with their own mitochondrial proteins. Also, it has an inner membrane space in between the membranes, as well as a matrix. These are known as the major sites of energy production. Next, I wanted to talk to you about the mitochondrial dynamics. So this basically means that the mitochondria can fizz or fuse, fizz as in separate and fuse as in join. Here on the right, you can see a hyperfizzed cell. So all the mitochondria are separated, and some of the proteins involved in that are Drp1, which is the main protein we're talking about today, and it is recruited by these outer mitochondrial membrane proteins known as MFF1, FIZ1, and MID4951. And here's a little video of a real-time fission fusion events throughout a normal cell. Here you can see some fission events happening. Should happen a little bit. Yeah, that right there shows a little mitochondria fizzing away from the cell. And opposite to fission is this fusion dynamic, which is, like I said before, just mitochondrial joining. And the membrane proteins associated with that are the outer mitochondrial membrane proteins MFM1 and 2, as well as the inner mitochondrial membrane proteins OPA1. And you can see a little fusion event happening down here with a bunch of mitochondria fusing into a little ball. Next up, I want to talk about DRP1. It is known as the main regulator of mitochondrial fission and it does this by hydrolyzing GTP. So first it is recruited by those outer mitochondrial membrane proteins that I mentioned before. It is then phosphorylated by the cell cycle regulator CDK1 and cyclin B at the serine 616 position. Once it is phosphorylated, it is then activated, and when activated, it will wrap itself around the mitochondria and start hydrolyzing GTP. After every round of hydrolysis, the DRP1 proteins will further constrict the mitochondria until it is separated. And then the last bit of background to know is what are cancer stem cells. They're known as a subpopulation of cancer cells with the main ability to create tumors. It can create a tumor from a single cell, and it can do this by these other two abilities it has, which are to self-renew and to differentiate. So self-renewal is basically the ability for a cancer stem cell, once it replicates its daughter cells, will retain its stemness. And as well, it can also differentiate into whatever cell the tumor may need to grow and maintain itself. And that brings us into that in certain tumors, these are the cells that are known to have a dependence on mitochondria. And normally, these kind of cells don't, but in certain tumors, they do. Which brings us into the aims of our research, which focus on understanding the DRP1-driven mitochondrial dynamics in cancer stem cell enriched cell populations. Here's how we achieve that. So the way we achieve that is by transfecting our cells with a DRP1 knockdown and overexpression construct using lipofectamine 3000. We also, the cells we used are HACAT, otherwise known as human keratinocyte cells. We transform those with TCDD. And one population of note is our cancer stem cell enriched population, which is the TF1 that you'll see later on. We also used standard genetic manipulation techniques to knock down or overexpress Drp1 and its mutants. To gather our data, we used immunofluorescence, live cell staining, and confocal microscopy. So when using our immunofluorescence, we used the Drp1 and phospho-Drp1 primary antibodies. When live cell staining, we used MitoTracker green to fluorescently label our mitochondria. And our confocal microscope was the Zeiss LSM 700. And the concept behind confocal microscopy is basically using a laser to focus at a specific spot on the sample. Once it is focused, it will excite labels found or localized near the mitochondria. This produces a signal that is then captured by a light detector, and that light detector transmits it to the computer, allowing for the computer to generate an image. Lastly is our image analysis. So we used Mitograph version 2.1 to derive our fission fusion metrics. We also use ZenBlue, ZenBlack, and ImageJ for image processing, as well as Excel for data analysis. Down here you can see, here's an image from Mitograph, and here are some of the images we were using to put into Mitograph. Something of note, I also created some Excel scripts using the Excel coding language to speed up the workflow a little bit onto the results we gathered. So the first thing I want to take your attention to is this Western blot. You can see that we used two different types of knockdowns. We used a weak knockdown, as indicated by this lower DRP1 expression, as well as a strong knockdown, which almost has no DRP1 expression. The next thing I want to take you to is this gene expression profile. This is basically just showing that for different gene expressions, the DRP1 weak knockdown and the TF1, our cancer stem cell enriched line, they follow the same direction. As well as the strong knockdown does not correlate at all with either the weak knockdown or the TF1. So that brings us to our correlation graphs. On the x-axis is the fusion metric, on the y-axis is the fission metric. Here's a little reminder of what that might look like. And so the first thing you'll notice are these red boxes. These represent what we've termed as a Goldilocks zone for cancer stem cells. And that basically means that in terms of DRP1 expression, cancer stem cells like to keep this range of fission and fusion. So they typically reside in this box. And as you can see in our cancer stem cell enriched line, a lot of the cells are occupying this box, as well as in the knockdown, the weak knockdown, at least compared to their extremes, which do not occupy this box too much, as well as their controls. Next, I wanted to talk to you about the activation of DRP1. So here you can see in the total DRP1, the control has a lot, then there's the weak at an intermediate level, and then strong at a even weaker and an even lower level of DRP1. Next up, what you'll notice is that in the phospho-DRP1, about half of the controlled is phosphorylated, while in the weak knockdown, almost all of it is phosphorylated, and in the strong knockdown, you'll notice that even more is phosphorylated, and this is further substantiated by this ratio. And lastly, I wanted to talk to you about these phosphorylation graphs. So the first thing you'll notice is that with this Western blot, is that TF1 has less phospho-DRP1 than parental, but it has more DRP1 in it. That's just something we'll get onto later. The first thing I wanted to bring your attention to in this fission graph is that, first I should explain, the purple represents a phosphomimetic phenotype, as in the DRP1 is always active. The brown represents a phosphodead phenotype, as in the DRP1 is always inactive. The green is a wild type, and the blue is a control. Here you can see that the phospho-DRP1, or the DRP1 constructs, they work, as in the phosphomimetic shows higher fission, as well as lower fusion in the parental. However, something of interest to us is that in the TF1, which we already know has less, or has more DRP1, when we phosphorylate all of them in this phosphomimetic construct, you'll notice that this the fusion actually increases, and we found that to be statistically significant as well. And that brings us into our conclusion, where, as you can see, we came up with the Goldilocks zone for that, basically showing that a parental cell with fine-tuned DRP1 repression enters the Goldilocks zone, and then once it is transformed by TCDD, it becomes a cancer stem cell enriched line transformed by monocarcinogenic stress. As well, this superoptimal DRP1 repression can also be transformed by TCDD and enter into the cancer stem cell enriched line. Here are my references. Are there any questions?
Well, first of all, it's a very interesting presentation and research that you're doing. So yeah, any questions from our audience or from our judges? Don't want to step on anyone. You can put it in the chat message as well, if that's easier for you. While we're waiting maybe for someone to ask, I'll ask a quick one that I've been asking each of the presenters, just because I'm very interested in how people have overcome the challenges. So, you know, what challenges did you have this semester conducting this research during COVID?
Well, for me, it mostly had to do with not being able to be in the lab over the summer, because before that we were about to choose what my project was going to be, but then COVID happened and we weren't able to do that. So, I mean, if we were, that would have most likely ended up in an entirely different presentation.
Gotcha. And I think that kind of connects to Destiny's question that was put in the chat. You know, what made you choose this topic?
It just flowed really well into what our lab was going towards. As well, I wanted to make sure that I became an author on a paper before I left UAB for grad school, just to hopefully give myself higher chances. That's the main thing, is that it just flowed really well into it. We're about to release our paper soon.
Fantastic. Another question in the chat, what are the clinical implications of this system?
So I believe that using or figuring out what the DRP1 Goldilocks zone implies is that we can hopefully tackle specific tumors. Unfortunately, this doesn't apply to all of them. This is more of an exception to the rule. However, the tumors we can come across that use this mechanism, we can hopefully target it and give people like a DRP1 inhibitor type of drug or something like that. And that'll hopefully lead to easier eradication of these types of tumors.
Excellent. Any other questions or comments from our judges? Some compliments from Adam. Thanks for presenting. Good job. I second that.
Thank you.
I think it's one you'll want to share with family and friends, hopefully. Anything else before we stop our recording? Just want to give everyone an opportunity to chime in.
Great presentation, Alan. Sorry, I'm running.
Thanks.
You represented the lab really well.
Thanks.
Awesome. Thank you for joining us, Dr. Spurlock.